All right, it looks like people entering are slowing down, so we'll get started. Thank you everyone for coming to the second webinar in this webinar series for the OCB DNA intercalibration. I'm Katie Roach. I'm one of the co-organizers for the Metabarcoding Intercalibration, and today we're going to hear from two other intercalibration efforts, one focused on zooplankton DNA metabarcoding and one focused on metatranscriptomics of marine microbes. So we're going to start with Leo Blanco Bercial, who is at Arizona State University, Bermuda Institute of Ocean Sciences, to talk about the DNA intercalibration for zooplankton. Go ahead, Leo.
All right. Thank you very much for the invite to Rob that is not here, also for all the everybody else in the team. All right. I hope that now you are seeing what I was expecting that you will see. And so again, thank you. I'm going to present some of the results that are not in the paper that came out of this experiment, intercalibration experiment. And that's why I call it the director's cut. It's all the little details that you cannot put literally on a paper because then will be like infinite long. So sorry, 1, 2. So this starts from a SCORE working group, the 157. The lead was Ann Buckling from University of Connecticut. 23 members, 19 countries. And the focus was about the 8,000 species that we have of holozoplankton, so animal plankton. About 15 phyla, 32 classes, highly diverse, not if you compare it to the other 95% of the eukaryotic diversity that are the protists. I know, but again, relatively high diversity. So all these animals are drifting. And we wanted to combine these molecular and morphological approaches that the team, had been working for a long time. What we wanted to do really was create an open access web portal for DNA barcodes for marine zooplankton. That is this MetaZooGene portal that we have, that we also develop databases to be able to use Chime or Data2 or Modur. That is what I use when you are doing all your metabarcoding approaches. We wanted to design an optimal DNA barcoding pipeline for marine zooplankton that we have developed long time for the census of marine zooplankton that started in 2005. So the last 22 years working on this still. And then develop these best practices for DNA metabarcoding of marine zooplankton biodiversity that many groups, like your group, is also trying to approach. Like, how can we do this on a homogeneous way that is better for everybody? So then Anne wanted to do this intercalibration experiment. And I said, Anne, another one. Like, I've seen so many papers. Are we really going to do a new one? She did say yes. So, all right. So then we put together everybody in the team, North America, South Africa, Australia, Japan, Europe. So all continents, researchers from all continents. We came all together and we said, all right, let's take some samples that we have taken near our institution and let's share it with each other. And let's try to do some kind of experiment in which we can at least check some of these points in which we might have difference when you run a metabarcoding analysis. So Connecticut, BIOS, again, many countries in Europe, is a long list. So this is the map that I had with all the samples. I chose this way to represent the oceans, because if you put in a regular map, sometimes I felt like I was not representing properly or the Pacific Ocean. But one day I put it in this kind of representation, and you have here the Southeast Pacific, the North Pacific, the Tasman area, the South Atlantic. This is IMT cruise, the Indian Ocean, but of course, when I'm working in the Gulf of Maine from Northwest Atlantic, part of the Eco Mountain series, and then 3 regions in Europe, the fjord, the Baltic, and a small intertidal plain in the North Sea. Okay, so there are very different environments, from hyperdiverse environments such as bats of these regions in the Northwest Pacific to some samples that should have very, very reduced diversity, such as the Baltic or this area in the North Sea, or the fjords, isn't it? So we wanted to not to have all this kind of like super variability between all the experiments. So the DNA extraction was done by each lab. So each lab took the sample that they prefer, and they did the DNA extraction. So then DNA extraction is not a variable. Okay. Then we created 11 aliquots of that extraction, keeping 2 for your own lab, because you should run your experiment at least twice, and then sent 9 other aliquots to each of the other labs. We actually sent it all to Connecticut, and in Connecticut redistributed everybody because it was going to be much cheaper in terms of saving shipping costs. And then after that, we told them, okay, we need to run B1, B2, B4, B9, CO1 with these primers, but we didn't give them the whole primer that is the primer itself plus the link plus no. So we just tell them your primer has to have this sequence. After that, we left them to have any kind of protocol that they wanted to have, like 1 PCR, 2 PCR, using Nextera, using kits, using all homemade style, etc. Sequencing facility, hiring somebody like I do. I do all the sequencing abroad, but some people do that in the lab because they have enough resources to have all the sequencing done at home. So then that part is the part that got really wild, very diverse. Isn't it? Like 150 PE, 250, 300, mixing different markers. That was very interesting. Each lab will receive the sequencing, they will have to demultiplex, and then send back to me that I will do all the bioinformatics. I use the bioinformatics that I usually run. I use Modur pipeline to single variants for the case of all the 18S markers. Just one base pair distance denoising without removal of any of the rare variants. So I have all the singletons and doubletons and everything there. And for CO1, I chose a 95% similarity to establish OTUs, operational taxonomic units, because this is what I've been doing. I did during my postdoc, and we decided it's needed with the barcoding of life that about 97% is the similarity for CO1 of within a species. Okay. I didn't put any kind of abundant threshold to remove reads. So this is really a really brute, very pure data set. The paper, if anybody wants to read it, meta-eukomics intercalibration. So if you want to see the big results are there, but for this talk, I don't want to go and tell you what is in the paper, because for that you can read the paper. So then if you're looking at ecological patterns, of course, like any other paper that have been published comparing metabarcoding approaches, they agree. So, but of course, B1, B2, B4, B9, CO1, they have a different variability. So again, from the very conserved region for the B9 to the highly divergent CO1, of course, you have then all these different, like B9 very conserved, B1, B2 a little bit more variable, B4 more variable, CO1 wildly variable. So then you have some differences due to these differences in these markers in terms of variability. We also notice that the higher is the local diversity, the lower is the reproducibility of these experiments. You have lower similarity. But the usual protocols that we use for metabarcoding, that is the rarefaction, build a distance matrix of some kind, the PCOA or MDS, etc. Actually, they are designed to be very robust against this kind of miscounting artifacts. They have they put a lot of weight in the most abundant ones, even after the transformation, etc. So then they are very robust against these things. And that's why we always find that they all agree. So here we have, for example, CO1, that is the most noisy of all markers. Here on the left, this is a regular MNDS, so multidimensional scaling, similarity between the different samples. Here in the middle is this cloud of samples where actually due to index hopping, cross-contamination, or something like that, that the once we remove them, each of the scholars was basically a region. And we find that, yes, each region is more similar to each other than to any other region. So basically, the laboratory is not a variable that really had a lot of weight on the analysis. But, I mean, it's not that we need metabarcoding to know this, isn't it? So is this enough for what we are trying to do? So I want to then, this is the part that is not in the paper. If you go into the details, if you go then into the big table of how many different single variants you have and the number of each of these variants that you have, you are going to start to see differences. So this is the distance matrix. I think I built this with V9. And something that you will realize if you take those numbers is that the replicates within laboratories, remember that I said that we will keep always at least two replicates of the sample and run it twice from your laboratory. So in this case, the replicates, for example, in the laboratory number, in the lab 4 for their own samples, they run 4 replicates. They have a higher similarity compared to all the other labs. So that means that having a different lab is going to have a difference in terms of the similarity. They're going to not to be that similar to each other, right? Here is one, for example, this group 5, the same, like 59%. 59% is higher than any other numbers, isn't it? It's not, for example, here you might have this 59 between 4 and 1, etc. But 5 compared to the others is always be more closer to itself than when you compare it to the others. And the difference is more clear when you have like CO1. This is when you have the big differences. If you go to V9, that is a very conserved marker, then the differences are not that high because, well, you don't have that much variability to get the differences, isn't it? And this is very important. For example, if you take you take V9 and you put which are the top 10 single variants of each marker for one sample, isn't it, between the different laboratories. And then V9, more or less, they will all agree. Maybe you will need like 12 always, like there will be a couple of those that will go up and down. But if you go to CO1, likely the top 10 for one lab, they might be spreading the 220 or top 30 if you go to a different lab, isn't it? So then if you are really looking for single variants for one group, then you are going to have differences between laboratories. And then there are some laboratories that, who knows why, they always show low affinity for every other. For example, laboratory number 9, that also was the one with the highest diversity, always saw very low affinity with the other laboratories. And that was consistent between the different markers. In the paper, there is this figure 6, in which I saw this is basically a rank between when a lab detected the highest diversity for a single sample and marker, isn't it? For example, in B1, B2, so the group, the laboratory number 9, always was the one getting the highest diversity from each sample. But actually, laboratory 9 did that for almost every single marker. And the other laboratory that was very good at detecting high diversity was the laboratory number 2. Because if this was random, what you would expect is that some people with some labs might be sometimes the first one, sometimes the 9th or the 10th. So we'll have more of a complete, full of small circles. However, we see that this is not random. Some laboratories always have higher diversity. In this case, were laboratories 2 and 9. And I start to look in depth, like what's going on here. Well, 2 and 9, they use the same, for example, the same tag. We use the New England Biolabs tag, the Q50. So then this is the point. Is this a real, or is it just random contamination that we're having, like crossing between different samples? And how can we differentiate between those contamination or real? At the same time, if you look here, these groups in the middle, these like 5, 6, 7, 8, they were always like detecting very low diversity. They were not even like in the middle group. So there was something in them also that I don't know why. I mean, I look at a lot of the data, a lot of the variables, but they always detected low diversity levels. This is also a figure from the paper. This is an astral tree. An astral tree, this is a tree that is based in other trees. We use this for phylogenomics. Okay, so instead of, in this case, instead of building a tree of genes, I'm building a tree based in trees that they were derived from the similarity matrices from each marker and each region. So this tree is based in 40 different trees that basically is all the different variables that we had in this lab. And so they put it together, and this is the support of the different nodes. If this had been completely random, we shouldn't be seeing this kind of supports of almost 100%. Okay, we should be seeing like lower support, like 60, 40, something like that, isn't it? However, we see here that groups 2 and 9, they usually have the very similar differences between the different samples, the same for 6 and 7. Okay, so then there is some kind of non-random component that is telling us that actually there are differences between the different labs. And how can we know if that is true or not? Sometimes it's very difficult. For example, in group 9 was the only group that detected Metridia curticauda. It's a small copepod, open ocean, in the Baltic Sea, here. So if you go to the Rasul's webpage, that is a French webpage where they put the distribution of all copepods in the world. I'm an expert in copepods, that's why I'm choosing copepod examples. So, well, Metridia curticauda is almost in all the oceans that are not well temperate and cold oceans, not in the Arctic or the Antarctica. So then it's possible that maybe a group was able to detect Metridia curticauda. It's not in very low numbers, but maybe, well, there's the possibility that a small egg or a copepodite ended up there, very low DNA level, and maybe the metabarcoding analysis carried up. So I cannot say that it's impossible. However, the other problem is that the same group detected Calanoides patagoniensis, that, as the name says, is typical from the Patagonia, so from South America. And this part, I can be very sure and say, look, you cannot have Calanoides patagoniensis in the Baltic. You had some kind of cross-contamination between the samples. The funny part, the negative controls were always negative. Apart from one group that they use NextSeq, and actually the laboratory that ran it external didn't want to send that, or they said that they failed to produce the negatives. There was a lot of index hopping. This big cloud of samples that looked like have contamination, actually they were done, there was the metabarcoding done with NextSeq. Okay, so, but all the other negative controls, I checked them, they were all negative. But there's still several groups that got this cross-contamination. So we have to be very careful with that. Positive controls. I think we didn't have like artificial positive control, and I think we should have used them and think them very carefully. I will go more about that in a couple of slides. Because there is going to be always a small leak from sample to sample, but which is the limit? So in NetSeq, in which we had a wild index hopping, I will go about that later. The first leaked single variant, that is the first cross-contamination, was actually in the top 20 in abundance. That means that anything that was below, despite that might be true, it might be mixed actually in this with the cross-contaminated individuals. In my SIG, the first leaked from other samples, the first contamination that I was able to detect, it was about in the top 400. It's in the 300, 500 region is when you start to see some of these leaking samples from leaking single variants from other samples. The problem is that they are leaked but surrounded by true variants. So what can we do? I was able to identify Calanoides patagoniensis, or in this case, since we have so many samples that were replicates from each other, okay, so this one, this NextSeq, this is the only one with it, the others don't have it. So this is a leak from this other sample that I could detect very easily. But this is my case that we are doing replicates of the same sample, and this is a control experiment. I don't know how we will be able to do it if it's not a real experiment. And again, what do we do? Do we remove everything after the leaked one, after the contamination? That will remove a lot, like 90% of the individuals, of the taxa you have there, isn't it? And of course, it's going to depend on the diversity of the renal sample. The ones with low diversity, if you have a sample with low diversity, the leak one is going to be much higher in the rank. With high diversity, it's going to stay like hiding below, so it's going to be even more difficult to detect. So then you have contamination. Again, do not underestimate the problem now with we had a problem that we were using multi-platform. So through the classic Illumina with 4 colors together with the NextSeq that has the 2-color chemistry, blue, green, or no color, or blue and green for the 4 bases. And the flow cells, they are patterned, so they have these wells on it. And that actually increases the index hopping that they call it. And so in NextSeq, what we detected is that some of these jump single variants, sometimes they were up to 5% of the total number of reads on the contaminated library. Because that depends actually on the library load and things like that, when you are actually preparing the chip. So that part is very difficult to control. And of course, you have this problem that with the new versions, for example, with the new Illumina, that also uses this 2-color chemistry and the patterned flow cells, we are having this much higher diversity levels, because I'm running a different experiment now with some colleagues, not to publish, just internal, because we have to switch from classic Illumina to something else. And what we detect is that our diversity is much higher using the new Illumina MySeq than with the old one. And the Illumina rep is telling me that it's because they are much better at characterizing the rare biosphere. But actually, I think it's they are pure higher error rate and index hopping. See, my opinion is that characterizing the rare biosphere is something that you do at the moment of PCR and etc., not when sequencing. Okay, and as of today, I don't think that we can really solve this issue. And please always use some communities of taxa not present on the samples. And one final part before I go into what I will do if I were you is that something that we have to consider when we were doing this cross-calibration experiment is that we allow them people to use any kind of length of sequence and they chose. So some of us chose like 250, 150, 300 per end. And when you try to put them together, things like this happen. So this graph is comparing the raw reads per sample compared to the reads that pays quality control. And in yellow is the raw data, non-treated. And these two clouds, there are two groups that instead of using the 250 PE, they use the 300 or the 150 base pair. It was a very small fragment, P9. So I had to do a different pretreatment to both to be able then to put them all in the red. That is the once I pretreat, everybody is same. So I was able to do it, but because we have only like 40 different groups. But can everybody do it if you're going to look at thousands of different runs, for example? So if I were you, just to finish two slides, what I will be thinking about, the first part is using a mock community of taxa that cannot be present in the samples. Something is similar to what you will get, but impossible to have. Nothing that can be food. I can tell you a lot about tilapia and salmon present in the Sargasso Sea from my eDNA samples. Obviously, they are not there. It's what they eat in the boat. So nothing that can be used for the biomedical industry, like cow and pig, etc., that is in all the buffers that we use. And we try to put a diverse groups, trying to cover all the phylogenetic breadth that we are using. For animals, will be easy, like insects, earthworms, squirrels, non-commercial fish. But I don't know how you can do this for this when you're trying to do eukaryotic protists. Like it's impossible just trying to get that kind of diversity into a mock community. And trying again to make it hyper-diverse, which is literally impossible. And the other part I would think about is about that MySeq, classic MySeq 4-color Illumina is dead. It's not supported. And this MySeq I100 is the iSeq. So similar to the NextSeq and the NovaSeq is based in a 2-color chemistry with a pattern and flow cell. They being the quality scores instead of giving you this 1 to 30. It's not a big issue, but I don't think that the other two or Chime is able to do it yet. And the error rates and the tag jumping, despite they say very low, at least in the ones that have been tested independently, the NextSeq, etc., they are much higher actually compared to what was happening in the classic MySeq. Also, they need unique barcodes, so you cannot use this combinatorial approach of 8 forward and 12 reverse. Each sample has to have their own forward and reverse barcodes that are different to everyone else. This is about $20,000 investment per marker. That is actually pretty scary. And again, my experience is that still you get many more ASVs than with classic MySeq, but I don't know if that's real. I suppose that it's just this effect of index hopping. Some people suggest me to do a strong deblur settings, but then you lose all the rare. Again, if you are just interested in big ecological patterns, it will be fine. But if you are looking for which are these small guys driving some of the changes or linked to increases in temperature, then you might be losing those if you try to clear the samples that way. And I don't want to see my time. I think I stated, I'm sorry. I would like just to appreciate then our funders, the SCORE and NSF, and thank you very much for listening.
Thank you so much, Leo. We have a few minutes for questions. If anyone wants to raise their hand, or feel free to put questions in the chat. And while you're thinking about your question, I'm going to put the link for our next webinar in the chat as well. So feel free to register for that. That'll be September 22nd at 1 p.m. And I think I see Jed's hand. Go ahead, Jed.
Hi, that's fantastic. I really appreciated that. We also had exactly the same experience with 5% index hopping with the new platforms, and we're told we'd have to switch to unique dual indexes to use the Illumina platforms. which we did for some samples with like libraries that are already made or ready to make, because we make our own libraries. Instead, we found that AVIDI sequencing allows you to use the old combinatorial primers, and we get very good quality, and we're very happy with it. We're using it at University of Minnesota. Uh, so that saved us all that money of, we had like 20 by 20. We had a lot.
Yeah.
It would have been a very big expense to go to unique indexes. So I'm glad you also came up with ideas about how to get around it, but we threw out the samples that had the index hopping because we just didn't trust them.
Yeah, so I'm part of a different group again that we're doing mostly bacterioplankton and protists, and we have a 30 years time series. And now what do we do now? So we're going to do just the 5 last years that we need until 2026. And the next step, I told them, like, do we need to still do in Illumina? If this is the case, if it changed from 4-color to 2-color, is this big, we shouldn't be bound to continue with Illumina and try other platforms like Aviti. Yeah, might be the solution.
Are there any more questions for Leo? I think we have another minute or two. Or if you think of something else, feel free to put it in the chat and then maybe Leo can respond. We'll move on to our next talk, which is going to be about a metatranscriptomics intercalibration effort. And today we have two speakers, Natalie Cohen from the Skidaway Institute of Oceanography at the University of Georgia, and Sarah Hu from Texas A&M.
Okay, hello everyone. Can you see my slides? Thumbs up. Okay. Hey everyone, I am Natalie. I am joined today with Sarah Hu. We are two of the four members of the organizing committee of meta-eukomics, along with Adrienne Marchetti and Harriet Alexander, and you'll see a bunch of her figures today. We're going to walk you through insights gained from our microeukaryote metatranscriptomics intercomparison effort. We'll talk to you about the rationale for this working group, our experimental design. We'll share some results and some lessons learned. So we are a group of 37 individuals. So the results that you'll see reflect the hard work and contributions of all of these people. We are mainly a domestic US working group, and we had 2 international participants. So our interest in starting this initiative was twofold. There has been these large organized field efforts to increase sequence data sets across the global ocean. Some of those are listed here, like BioGoShip, Tara, BioGeotraces, and Atlantico. Some of these are completed or in process or in the planning stages. And these groups have done a great job putting together consistent workflows. So DNA and RNA is extracted the same way, and they use similar sequencing pipelines. But how can we compare products across these different projects? And how do the various methodological decisions that one makes impact the products that you get out and your downstream biological interpretations? Along those same lines, there's an increase in the number of individually produced sequence datasets. This is a map showing the number of NCBI SRA entries matching metagenomic or metatranscriptomic library source search terms. And these should be marine samples. They have marine keywords somewhere under their biosample attributes. So there's lots of these produced by different labs, presumably using different methods. So how does that impact our ability to compare and draw biological results? There's also BioGeoscapes, which is in its planning stages and will involve international partners using their own workflows across labs and, of course, across different timescales. So what will be most important to consider when comparing those products across expeditions? That's some of what we hope to address. We drew inspiration from previous intercomparison efforts. There was the OCB organized Ocean Nucleic Acids Intercalibration and Standardization Workshop. This brought experts together to think about sources of variation and important factors in intercomparison. They talked not only about eukaryotes, which are our focus, but also for prokaryotes and viruses. And in terms of the tools, not just metatranscriptomics, but factors to consider for metagenomics and metabarcoding. Their workshop report is available online and was helpful for us to consult. We also drew inspiration from the OCB Ocean Metaproteomics Intercomparison study. They had both a wet lab portion of their comparison and then an informatic portion. And we found that really helpful too. And we modeled our experimental design after theirs. Their work was published just a few years ago in Biogeosciences. Our goals were to perform an intercomparison of eukaryotic metatranscriptomes, focusing on how RNA extraction procedures differed by lab. Our envisioned goal is an article summarizing these results and best practices that we put forth back to the community for those interested in performing their own eukaryotic metatranscriptomic studies. We also had broader community-wide goals. We wanted to better understand how our workflows impact our understanding of marine microbial diversity and function. We wanted to better understand the utility and limitations of our current methods, their sources of variability, and we wanted to share our results with the broader environmental omic community beyond aquatic sciences. There may be groups working in other systems that are curious how different extraction methods may lead to the loss or enrichment of certain groups, or how library prep factors can influence the sequence content downstream. For those of you unfamiliar with metatranscriptomics, I'll walk you through what a workflow typically looks like and the different sources of variability that can be important in giving you different RNA pools at the very end of this. So for oceanographers, a lot of us collect our samples offshore on ships. And so the first decision you make is how are you going to collect that biomass? Some groups use underwater pumps, which is what we used, but maybe more commonly people use CTD Niskin bottles. So how do you filter that water? Do you use vacuum filtration or peristaltic pumps? What is the volume of seawater? How long do you filter for? Filter types and preservations. All of this may make a difference. For our intercomparison, we used one collection method. So we really start at step 2, which is the extraction step. This includes sample handling, the kit used or reagents used, whether you break open cells and how you do that, and if you're removing DNA to get pure RNA. Then we have our library preparation step. Commonly, we do poly-A selection for the eukaryotes, but also popular is rRNA removal, which will leave behind mRNA from both eukaryotes and prokaryotes. Then we have the sequencing used, so the platform, which will dictate how long your reads are and your throughput. Then we have all of our informatic steps. When you get those sequences back, you can remove contaminants, you can trim low-quality regions. Many groups perform de novo assemblies, and if so, the assembler that you use can matter. And some groups direct map back to references. Then we have our annotations, which include clustering orthologs, predicting proteins, and the types of taxonomic and functional databases used. And then we also have read mapping and normalization. So we are definitely interested in these informatic steps, but we decided early on to just focus on the wet lab extraction methods, which is what we'll talk about today. Our biomass was collected using clean pumps, and we worked with Max Sato's group. He had a cruise happening in the Costa Rica upwelling region in May of 2023. He generously agreed to collect biomass for us. This closely mirrors what they used for their metaproteomic intercomparison. These McLean pumps are battery operated, they're attached to the line, and they sit at a certain depth. For us, it was 30 to 35 meters. They filtered 100 to 140 liters onto those filters. We captured the 0.2 to 51 micron fraction. These 4 pump heads all had a filter on them. So it was 142 millimeters, and we were able to get a lot of material on that. So this is Mac's group that's sectioning those filters back in the lab on the ship. That's Paloma and Emily. So we had 1/8 of filters that we collected from these pump heads. Our work plan looked something like this. We sent 1 or more filter slices to each participating lab. We asked them to perform their RNA extraction method using whatever protocol they would normally use. We didn't want to limit them or give them guidelines. We wanted the full extent of variability possible at this step. They sent that RNA back to Harriet, and her lab sequenced a set at 1 sequencing facility using the same kind of sequencing. So they were all poly-A selected, and we generated about 40 million reads per sample, and they were 100 base pairs long paired end reads. So that was 19 of our intercomparison samples. With the other filter, groups were encouraged to extract and then sequence externally, and we ended up getting a range of depths. So on the lower end, we had a sample that was 10 million reads. At the higher end was 360 million reads, which was great to show us that full variability. All of these were longer, 150 base pairs long. Some were poly selected, some were rRNA depleted, and that was 11 of our intercomparison samples. It was important to now use one consistent bioinformatic workflow because we just wanted to isolate and look at the variation due to those variables that we previously mentioned. We used the euchromatic metatranscriptomic pipeline that was developed by Ariana Krinos and Harriet Alexander. Both of them processed our 30 samples through that workflow. And here are the details for those that are interested. We produced 30 individual assemblies, each with their own taxonomic and functional composition and their own read count info. And relevant for today, we use the MMETSP database for taxonomy that had marine bacteria included. We came together as a group in person in Savannah for a 4-day hackathon where we worked with these data products. And we addressed these questions of how different lab protocols influenced assembly metrics, recovered content, and taxonomic and functional composition. Our participants were invited or selected based on their expertise, as well as career level. We did a bit of planning beforehand and we split people into groups so people knew what they were going to be working on roughly. It was important for all externally sequenced samples to be shared with us ahead of time because the Alexander lab had to make sure everything was processed through that bioinformatic pipeline ahead of the meeting. And then we had to transfer products from WHOI's cluster to UGA's cluster so people could physically interact with it. People also had homework. We asked everyone to bring a detailed wet lab protocol with as many details as possible, and the metadata group had the important job of consolidating all of that information. And as I mentioned, we are interested in how bioinformatics influences things, and so we did have a secondary informatic intercomparison happening at the meeting, but we subsequently decided to just focus on the bigger wet lab extraction. I'll pass it to Sarah now for some results.
Thank you, Natalie. So this is like the bird's eye view of the massive amount of metadata that was collected, and it's still kind of being curated. So from the metadata group, they collected all this information from each of the participants. So each row is going to be a slice of a filter from a lab group. And it was important, of course, to get all of this information so we can start to understand if some of the outcomes that we get downstream in the pipeline from the wet lab, if it's linked to anything upstream, like Leah mentioned about the TAC polymerase used. You can do the next slide. So this is a breakdown of what this large figure has. We have different extraction variability here. One of the more popular ones right in the middle with most of the lines, I'm realizing you can't see my arrow, has the QIAGEN EASY, which is often used for the RNA EASY protocol. We have beads. Things were labs varied in what bead sizes they used, or if they used one bead size or multiple bead sizes, and how long they did a bead beating step to lyse cells. And we've also captured concentration and RNA integrity number, RIN number. We had a range of sequencing centers, like Natalie mentioned, and technologies as well. And on the far right, we have some of the base sample statistics, starting with BOOSCO completeness and the kind of the core preliminary QC steps run on our output data. You can go to the next slide. I've split this figure in two to kind of show a couple of examples that I want to pull out. On the top in the red, sample 58 here is one that we had a lower concentration of, actually, I think it's supposed to be highlighting the one below it. No. But we had a RIN number of only 5 in the top row, and then in the second row of this figure down below, we have really high BOOSCO completion, and then really high total reads, and that subsequently went on to mean like higher transcripts of like that were greater than 500, 1,000 base pairs. But on the next slide, I have another example that I'm pulling out where the concentration was higher than this other one, RIN number was higher, but the total number of reads was a lot lower. despite now on the bottom row in the purple, this sample 53 ended up having the longest contig included and a high mean assembly length. And so the part of like Natalie mentioned, we're in like a synthesis right now of putting this manuscript together, and we're going to be kind of getting into the detailed nitty-gritty of what factors in the wet lab step might contribute to some of these differences. You can go to the next slide. So this talk is kind of a the end. We're going to talk about some lessons learned, and this is one thing that kind of happened from the hackathon to where we are with the progression of data from the hackathon to getting things refined for the manuscript. So Katie was involved in producing this figure after the hackathon, but the metadata group compiled all this data, and these were some of the ideas for figures, or we had preliminary figures that had NMDS plots like on the left. And both these figures are showing the same symbols and colors, where we have poly A tail selected libraries and circles, and the triangles are showing RNA depleted libraries, right? And the sequence length is the pink color is the 100 base pairs, and the blue color is 150 base pairs. And so we're seeing that this progression of figures as we've continued to refine the data and add some samples along the way, and that both read length and library prep make quite a difference in the number of proteins that we end up with at the end. You can go to the next slide. Yeah. And the next couple of slides will focus on taxonomy, some of the taxonomy results that we got. In this case, so again, focusing on the library prep steps here, we have the yellow circles showing the rRNA depleted libraries, where you're going to be left still with messenger RNA from both eukaryotes and prokaryotes. And then the blue slices have poly A tail selected libraries, where the lighter blue ones are all from the same group that when we sent out samples for sequencing at Columbia. And the main thing here is that the rRNA depleted libraries do have a lot of the prokaryotic contribution, and the y-axis showing the proportion of transcripts per million. On the next slide, we've taken out the prokaryotes, and so things get reorganized here, right? Because the idea with the rRNA depletion is that maybe one would want to both look at the prokaryotic component and the eukaryotic component. So if you were to do that, what does your data look like in comparison to those that are maybe doing the poly A tail selection to isolate the eukaryotic messenger RNA? And we're seeing still some differences in the relative abundance of that RNA. So where the rectangle is shows the poly A tail selection kind of increasing, having a higher number of certain chlorophyte and green algae. And then we have the RNA depleted one having a little fewer of those, and a lower relative TPM of the other category, and some of the dinophysis that we would assume would be pretty high, would have a pretty high relative abundance. But this is showcasing some of the differences we're seeing, and I think that library prep step does, cause, or does, change some of the taxonomy, the relative abundance at least. But regardless of everything, if you kind of take this in as like presence absence, we're seeing a lot of the taxonomy is mainly consistent, regardless of the wet lab protocol. The other thing to the right of this black triangle is showing one group that one slice of the filter extracted and sequenced that has a really high abundance of this lighter green chloropictophyceae group, and compared to everything else. And so I think that we are still kind of working on kind of tracking down what variables in the wet lab protocol might produce a pattern like this. Another thing, if you want to go to the next slide, another thing has to do with this group on the far left of this figure. And here, the first two, on the leftmost side of this is showing higher relative abundances of primarily this dinophyceae group in the blue colors, and super low of some of the other taxa detected. And upon looking at those samples after the hackathon and trying to figure out what was going on with them, these ended up being a subset of filter slices that were taken 5 meters away from the others. Okay, and so in order to determine if this was a result of a sample being 5 meters in a different direction, so the rest of the samples were from 35 meters, the sample, a third from the left, 061, was resequenced later on, which came from the 30 meters. And I think that ended up having very few contigs and like poor quality reads overall, but we did recover more of the taxonomic signature that we would expect from the other samples. And so again, in terms of thinking about intercalibration, we needed to go back and resequence stuff to kind of fill in some of those gaps to kind of understand why we saw certain patterns in the differences in taxonomy. But I think it was also relieving as a group to see that the taxonomy was largely consistent regardless of these wet lab protocols. Okay, next slide. Another thing that, especially when we start thinking about the bigger scale, like ecological interpretations of our results, we wanted to pair this with other groups. And so the samples from our intercalibration are all from where this the star is, and then the other samples in gray were kind of chosen as outgroup comparison samples from other places in the Pacific. And so when everything was classified to the class level, everything was placed at the class level, just using eukaryotes. This is when we used, and the different library types are shown by symbols again, using the same color for the RNA depleted one, and then the blues are showing the poly A tail isolation. And SRA in the gray triangles is showing the outgroups, right? And so we're seeing that when we get all the data, reassemble it all together, map to individual assemblies, the products of our intercalibration that are all polytail selected are in more agreement than the ones in outgroup, right? So we're seeing different patterns starting to show us that we have more agreement in our samples, regardless of the different methods that we used. Yeah, we can go to the next slide, I think. Okay. So I showed just a snippet of some of our results. Like Natalie mentioned, we've been focused on a lot of the wet lab methods, even though we did also experiment with like, each lab group doing some of their own computational, their individual computational pipelines. Our main things so far have shown that the sequence depth and read length really, really matter. And especially when it comes to kind of the end product of having more higher number of predicted proteins. The taxonomy was largely consistent across lab groups. There was a big difference between the poly-A and rRNA depleted taxa, or RNA depleted library prep protocols. And I think that one purpose that these processes serve is to really think about if you are going to go do a meta-transcriptome study, design it from scratch, or you want to compare your results to someone else's group that has done a different library prep. This is kind of a lot of new information that you can use to kind of make the best choices for your comparison or your study. Then we also looked at, we're still kind of digging through how the relative abundances of certain taxa, if it's related to changes in the depth layers, lab groups, or even the species-specific, how cells might lyse differently as well. So that's another thing that we need to start considering. But regardless, we were happy to see that the taxonomic composition is more consistent with internally than with the other metatranscriptomes elsewhere in the Pacific. You can go to the next slide. So as we think about the project as a whole, then this is going to be part of our manuscript as well, but it was helpful to have like really solid communication. OCB helped a lot with that. And we needed to have good points of contact for each lab group. so we can get feedback back. If you look really closely at my massive metadata table, there's still a couple things that we're ironing out and we're finding that we need to go back and maybe add another layer of information to the heat map. And continuing to strive for like monthly updates was helpful to kind of maintain momentum on the project because we kept having to reprocess data, collect more information. And the other thing that was really apparent during the hackathon was that it was a great community building process. I think everyone met a lot of new people through that hackathon process. It also presented an opportunity to do more cross courses in bioinformatics and also trying to figure out how to arrange the group so it built on people's strengths and having the individual groups. And I think that, and it's exciting that a lot of the main ideas and preliminary messages that were conceived at that meeting have kind of are maintained in the current manuscript, and we'll kind of have structured that. Would definitely recommend the required homework ahead of time and the solid setup, delegation of tasks and assigned roles for in-person hackathons like that. So like I mentioned, I think it was really exciting to have preliminary results kind of appear in real time when everyone's in a room together in breakout groups by topic and then coming together. And I think that as we continue processing this data and we're working on finishing up the manuscript, we're seeing that our results have continued to change a little bit based on Leo actually mentioned it, and Jed mentioned it with the follow-up question of like maybe there's going to be changes in sequencing methods moving forward, but now we have even more information to kind of figure out how to deal with those changes coming up. Next slide. Do you want to talk about timeline?
Sure.
Yes.
So we have a timeline here just to give you an idea of how long it took from like inception to products. So Harriet had the idea for this working group after we saw BioGeoscape's webinar back in 2023. Her and I sent feelers out to others to see if there was interest in such an activity, and there was, which was great. Mac was able to collect biomass for us on one of his cruises that were coming up. And then we hit the ground running. When those filters came back, Harriet's lab distributed them. Then people started their extractions. In that time, we submitted the OCB proposal and got it funded, which allowed for our in-person meeting. And that allowed people to sequence externally. The Alexander lab sequenced our consistent set. And then by early fall 2024, we were getting ready for our in-person hackathon, which happened in October. And then in the months after that, we were finalizing results from that meeting and reaching out to participants on that. Harriet presented a presentation with preliminary results at the March 2025 ASLO meeting. And then in the years since then, we've been refining. So as Sarah mentioned, we updated the algorithmic pipeline, we sequenced an additional sample, we've been making filters and drafting text. And so we're looking forward to sharing that draft with our working group members soon. So last piece of advice is to pace yourself because progress might be slow. Definitely take advantage of in-person gatherings. This was us just a few weeks ago now at the GRC meeting in marine microbes. And coming together is so important. This is Harriet blowing our minds with some updated figures that she made, and it's really valuable to like sit down together and make internal goals. And it is a community effort, right? Like we all have our own funded projects and cruises and classes that we teach. And so it's definitely community driven with the time and the resources that we have available. So we'd like to thank OCB for funding for our in-person meeting and formalizing us into a working group. We definitely used a lot of compute space, so WHOI's Poseidon, as well as UGA's cluster for our hackathon. UGA Skidaway Institute helped a lot with the logistics of the in-person meeting. And then funding for the sample distribution and sequencing itself came from Harriet through a Simons Foundation Award. And the members of our working group, they had to do those extractions and externally sequence on their own. So thank you to their funding sources. And Heather and May at OCB for their support throughout. And we will end there. Thank you.
Thank you so much, Natalie and Sarah, for that interesting presentation. We have a few minutes for questions. So again, please raise your hand if you have a question or pop it into the chat. And I will also put the registration link again for the next webinar, which is going to be Jed Furman and Tatiana Reinierson speaking about metabarcoding data and how we relate that to biomass using internal standards. So again, we have a few minutes for questions. Feel free to raise your hand or put them in the chat. And I don't see any questions yet, so I'll ask one to get the ball rolling. I think it's really interesting to see that the sequencing depth and read length played a big role in driving some of the differences between samples. And I wonder if you're able to approach recommendations for what's most ecologically meaningful from those differences. I know it's hard because there's so many different variables on all of those different samples, but is there a best sequencing depth or a best read length that we should be using?
Yeah, that's a great question. Something that we noticed early on was the longer read length, so 150 over 100 was giving us more predicted proteins downstream, which maybe is intuitive, but it was cool to see that, and that seemed like it was consistent. In terms of depth to go with, Harriet and Michal produced some rarefaction looking curves, and you guys can jump in if you want, but they looked at taxonomic bin completeness with sequencing depth by subsampling our very deeply sequenced sample. And what we were seeing was like 60 million reads seemed to give you a good estimate of taxonomic completeness that you were going to get. Like increasing all the way to 100 or 200 million reads wasn't giving you much more benefit for the extra sequencing depth. But if Harriet or Michal are here and you want to clarify, you're welcome to. And I think 40 to 60 is what people were generally using for their environmental data sets. So that was nice to see that 360 isn't needed because that would be ridiculous.
Sorry, 40 to 60 million reads per sample?
360 million reads was our most deeply sequenced sample. Yeah, it was like a whole lane of sequencing for one sample. We just tried it to see what would happen. But yeah, it was a lot.
No, that's a lot. That's because the in metabarcoding we do, of course, much shallower. So yeah, pretty
Harriet,
impressive.
I think Harriet mentioned it was 60 to 80 for those field communities. So that might be a good recommendation going forward.
Great, thank you so much. I don't see any other questions. So again, please register for our next webinar and thank you all for coming today. Thanks again to our speakers, Natalie, Sarah, and Leo. I'll give you another
Thank
round
you
of
for the
applause.
invitation.
Thank you.